Welcome to This Explainer. Today we're diving into literally science's ultimate detective story, how complex life chemically evolved from dead, non-living matter. Think about it for a second. Every living thing you know, you, me, the trees outside, we're all essentially highly organized, self-replicating chemical systems, right? Systems capable of Darwinian evolution. But how did that system actually start? I mean, we've got a pretty solid grip on Earth's geological history, and we know how life evolved once it finally took hold. But that first step? The precise derivation of the very first living cells from a random mix of simple components? That is a massive, fascinating, and totally unresolved puzzle. So we're going to walk through the exact steps, the chemical roadblocks, and the absolutely brilliant evolutionary workarounds the scientists have uncovered in the search for the origin of life.
We kind of have to start with Charles Darwin's famously romantic idea of a warm little pond, which really sets the stage for the chaotic, prebiotic Earth. Back in 1871, in a letter to his friend Joseph Hooker, Darwin threw out this speculation. He said maybe the original spark of life happened in some small, warm body of water, just loaded with ammonia and phosphoric salts. Add in some light, heat, or electricity, and boom, a protein compound chemically forms. Now, this was a profoundly insightful guess, way before we had anything resembling modern molecular biology. And honestly, it perfectly frames the environmental conditions that scientists are still investigating today.
Which brings us to the core mystery we're unraveling right now. We're charting the exact chemical roadblocks standing between a primordial soup and living, breathing organisms. Let's rewind. When our universe first began, the only chemical elements around were hydrogen, helium, and lithium. That's it. It took massive, short-lived stars to literally forge heavier elements like carbon, the absolute backbone of our biology, and then blast them across the cosmos in supernovas. But having those raw, dead elements just floating out in cold space, well, that's a whole lot different from a functioning, self-sustaining biological system, isn't it?
Let's anchor ourselves in deep time for a minute. Look at how incredibly fast our planet went from a hostile, heavily bombarded hellscape to actually hosting the earliest evidence of life. So Earth forms roughly 4.54 billion years ago. Very shortly after, about 4.48 billion years ago, a massive collision with a hypothetical planet-sized impactor named Theia created our moon. This impact stripped away our primary atmosphere and left us with a turbulent, magma-covered world. Yet, astonishingly, pretty much as soon as liquid oceans condensed, life took hold. Geologic records suggest life likely emerged between 4.32 and 3.48 billion years ago. I know billions of years sound like forever, but in geological terms, that transition was incredibly rapid. We're talking the absolute blink of an eye in the lifespan of a planet.
Now, for centuries, people believed in the ancient theory of spontaneous generation. They genuinely thought, if you just left some meat out on a table long enough, it would magically spawn flies and maggots out of thin air. It actually took careful observations from scientists like Francesco Redi, who proved maggots don't magically appear if you just keep the flies away, to finally put that myth to rest. Modern abiogenesis, though, it's entirely different. It proposes that the transition to living entities was absolutely not a single, magical, poof into existence event. Instead, it was a meticulous, slow process of increasing complexity. We're talking step-by-step chemical evolution from non-living matter, moving from molecular self-assembly to autocatalysis, and finally, cell membranes.
All right, let's get into part one, the primordial soup, the ingredients for life.
Early Earth was basically a completely messy outdoor chemical laboratory. Picture a potent mix of water vapor, nitrogen, and carbon dioxide just getting constantly battered by intense UV light and non-stop asteroid impacts. To get from that chaos to us, we need 4 specific chemical families. Every living thing functions through carbon chemistry and relies on these building blocks. Lipids to build the walls of our cells, carbohydrates like sugars for metabolism, amino acids to construct the protein enzymes that actually run our bodies, and nucleic acids like DNA and RNA to store and pass on hereditary information. A successful theory of the origin of life has to explain exactly where all of these came from without life already being there to make them.
And that is where the incredibly famous 1952 Miller-Urey experiment comes in. It gave us our first major clue. Miller and Urey essentially took a mixture of what they thought were early Earth gases, trapped them in a glass flask, and literally zapped them with artificial lightning. And miraculously, this kind of Frankenstein-esque experiment worked. It produced organic monomers spontaneously from inorganic gases, including amino acids. Since then, we've actually found amino acids inside meteorites and even in the star-forming regions of deep space. That proves that the basic building blocks of proteins are just naturally synthesizing all throughout the cosmos.
Okay, moving on to part 2.
the RNA world problem, which is a massive chemical roadblock.
Because, look, making amino acids is one thing, but creating the complex, self-replicating information polymers that are totally necessary for life, like RNA, presented a huge problem. See, it's widely believed that early life relied on an RNA world, where RNA acted as both the genetic blueprint and the catalyst for chemical reactions. But just looking at its building block, a nucleotide, shows you exactly why forming it is so tough. A nucleotide consists of a nucleobase, a ribose sugar, and a phosphate group. Prebiotic chemists have struggled for decades to synthesize this specific combination spontaneously. Why? Well, free phosphate was locked away tightly inside rocks, and those ribose sugars, they are incredibly difficult to synthesize robustly from scratch.
It gets even trickier when you look at the 4 canonical bases of RNA: adenine, cytosine, guanine, and uracil. Scientists realized that out of these 4, only adenine actually easily reacts with ribose in a prebiotic model. That makes the spontaneous formation of RNA incredibly difficult. If you dry and heat the other 3 bases with ribose, they literally just do not produce nucleosides. No reaction. And even for adenine, the yield is only about 2%. That is a massive roadblock. It strongly suggests that maybe the canonical RNA we know and use today wasn't actually the first player on the board.
So this leads us right into a major hurdle called the paradox of base pairing. Here's the issue. RNA bases only form stable bonds when they're already 4 or 5 units long in a strand. They absolutely do not form stable Watson-Crick base pairs when they are just single monomers floating around in water. So think about it. How could they possibly self-assemble and selectively pair up in a messy, chaotic primordial soup before they were joined into a long chain? Seriously, how could prebiotic reactions somehow anticipate that these specific canonical bases would eventually pair up so perfectly together if they don't pair as individual pieces?
Well, that brings us to part 3, the pre-RNA hypothesis, evolution before life.
To solve this giant paradox, NASA researchers suggest that RNA wasn't just some frozen accident that magically popped into existence. Instead, it was likely the product of chemical evolution. The idea here is that simpler, alternative informational molecules must have existed before RNA, acting as evolutionary stepping stones. Think of it this way. If RNA components like ribose and phosphate were just way too scarce, or if the bases couldn't pair up on their own, early life must have used chemical alternatives. It had to use things that did naturally pair up and self-assemble spontaneously under those harsh early Earth conditions.
And breakthroughs in prebiotic chemistry have actually shown us what that might look like. 2 alternative nucleobase analogs, cyanuric acid and TAPS, can do exactly what RNA couldn't. They can spontaneously self-assemble into these highly ordered hexads in water. It's amazing. Unlike the canonical RNA bases, which stubbornly refuse to pair up as single monomers, these alternative heterocycles easily find each other and bond together. This proves that the chaotic chemical mixtures on early Earth totally could have contained alternative molecules perfectly suited for mutual selection and organization. And it gets better. In a watery solution, these hexads naturally stack right on top of one another, forming these incredible, long, 3D non-covalent polymers. Once they're stacked up like coins, they could easily be stitched together by simple chemical bridges. That is so much easier to form and to recycle than the super rigid phosphate bonds we see in modern RNA. This brilliant workaround allowed these pre-RNA molecules to rapidly search through different genetic sequences to find functional enzymes. They were locally concentrating and organizing themselves long before complicated ribose backbones even existed.
Which sets the stage for part 4, our last universal common ancestor, or LUCA.
So, over time, those early chemical workarounds and pre-RNA polymers eventually gave way to RNA, and finally to DNA, which is much more chemically stable and allows life to store way larger genomes. This incredible evolutionary path leads us right to the doorstep of modern biology, LUCA, the last universal common ancestor. By looking at genomics, we know that all life on Earth today traces back to LUCA. We literally share 355 prokaryotic genes with it. LUCA was a single-celled organism, and it was incredibly sophisticated. It was anaerobic, meaning it survived without oxygen, and it was powered by a naturally occurring proton gradient near deep-sea hydrothermal vents. It already used the universal genetic code, DNA, and ribosomes. Interestingly, LUCA lived millions of years after the initial origin of life, acting as the ultimate evolutionary survivor of that crazy prebiotic era.
Recognizing that you, me, all of us, that we are the direct evolutionary descendants of those ancient, struggling chemical polymers, it leaves us with one incredibly profound question about our place in the cosmos. Earth is the only place we currently know of that harbors life, but the chemistry of carbon, of amino acids, and these self-assembling polymers, that operates universally. If the journey from dead rock to a primordial soup to living biology happened here through the natural, inevitable laws of chemistry, it really forces us to wonder, where else in the entire universe has this beautiful, chaotic chemical evolution already taken place? What other dormant mysteries are waiting to wake up? Just keep that in mind the next time you look up at the stars. Thanks so much for joining me on this explainer, and as always, keep exploring.